Good morning. I'm Tyler Martin, and here's the latest from Spectrum Autism Research. Mutations linked to autism may be detectable in men's sperm. By Emma Yasinski. About 1 in 15 men carries gene-damaging mutations, some with ties to autism or congenital heart disease, only in his sperm, a small study suggests. An advanced DNA sequencing technique detected the mosaic mutations among 25 men in the general population. Mosaic mutations arise after conception and thus occur in only a fraction of the body's cells. When you're looking at sperm mosaicism, you're right at the heart of what's going to happen in the next generation, says lead investigator Joseph Gleason, professor of neurosciences and pediatrics at the University of California, San Diego. Men accumulate mutations in their sperm as they age. Some researchers say these mutations explain why a man's chances of fathering an autistic child double between the ages of 30 and 50, whereas others suggest these mutations account for less than 10% of that rising likelihood. But the prevalence of the mutations detected in the new study remained constant over a 12-month period, suggesting that these genetic alterations arose when the men were embryos. The advanced sequencing method the team used can also calculate the likelihood that a child will inherit the mutations, odds that similarly remain stable over time. What they show is that mosaicism is a small but significant contributor to autism and congenital heart disease, says Anne Goriley, associate professor of human genetics at the University of Oxford in the United Kingdom, who was not involved in the study. And that analysis of sperm allows you to tackle this. Mutation mystery. Because many of the genetic mutations that contribute to a person's autism cannot be detected in their parents' blood, they are thought to arise spontaneously or de novo around the time of conception or in the parent's egg or sperm. In line with this idea, Gleason and other researchers have previously detected these de novo mutations in the sperm of fathers with autistic children. When the team presented their findings, though, we always felt like we got the same questions, says study investigator Martin Bruce, assistant professor of pediatrics clinical genetics and metabolism at the University of Colorado in Denver. Because men constantly produce new sperm, would a mutation identified in any given sperm sample still be there in subsequent samples? And would these mutations become more prevalent with age? To address those questions, the team took blood and sperm samples from 25 men up to 3 times over the course of 1 year. The men ranged from 18 to 70 years old. The researchers sequenced the entire genome of each sample at least 300 times, about 10 times more than traditional methods do. This deep sequencing enabled them to confidently distinguish mosaic mutations from sequencing errors. Without the advancing of the sequencing technology, it's almost impossible to find mosaic mutations, says study investigator Xiaozhu Yang, a postdoctoral researcher in Gleason's lab. The men carried an average of 33 mutations in their sperm, 2/3 of which are not detectable in their blood, the team found. The results appeared in Cell in August. Deep sequencing. The fraction of sperm containing these mutations remained the same for each man over time, and the sperm-only mutations were no more common among older men than younger ones. Taken together, this means that the mutations likely occurred during embryogenesis, the researchers say. By measuring the percent of cells with a specific potentially damaging mutation, we can actually make a prediction about the chance of having a child with the condition, says Gleason. If 5% of a man's sperm contain a mutation today, for example, that gives him a 5% chance of passing that mutation on to his children today, tomorrow, and 10 years from now. Of the mutations detected only in sperm, an average of 1.6 per sample occur in the coding regions of genes, and 0.3 are predicted to affect a gene's function, the researchers found. Such loss of function mutations in one copy of a gene appear in the sperm of about 7 out of 100 men, the team estimates. These mutations could account for about 15% of cases of autism, congenital heart disease, and severe pediatric conditions, they say. Of note, the study focused only on clonal mutations and not the more prevalent but less detectable non-clonal ones, which occur in only a single sperm. To have a full picture of disease risk for a particular individual's offspring, you also need to look at the non-clonal variants as well, says Maria Charor, assistant professor of genetics and neuroscience at the University of Texas Southwestern in Dallas, who was not involved in the study. Gleason says he and his colleagues hope to explore these variants in the future with larger sample sizes, he says. There's a whole other world we haven't even explored yet, which are these non-clonal mutations. That's all for today. Check back on Friday for more content from Spectrum Autism Research, or go to spectrumnews.org.